Assalamualaikum warahmatullahi wabarakatuh, and a very warm greetings to all of you. My name is Tuan Rafidah Binti Tuan Hassan. Would like to do my presentation about bioremediation of water in detention pond using Limnocharis Flava. Let's go to the introduction. Surface runoff from streets and parking areas brings oil, dirt, and pollutants. Every premises normally equipped with stormwater runoff treatment in order to protect surface water from pollution. Detention pond is a common stormwater treatment system and also used to help prevent flooding. In detention pond, water is treated through sedimentation and nutrient absorption. To review, generally, as is well known, clean water is an inevitable necessity in human life apart from food and shelter. With rapid growth in population and increasing industrial development in Malaysia, many water sources have become polluted. Moreover, water is a very critical natural resource for the world's fastest growing urban areas. Commercials, residentials, and industrial users already place considerable demands on cities' water resources and supply, which often require water treatment. Therefore, sustainable management of clean water and aquatic ecosystem requires eco-friendly and low-cost remediation techniques. Recent researchers have reported the potentials of aquatic plants for inorganic and organic pollutant removal. Phytoremediation technique is a branch of bioremediation that employs the application of plants for the remediation of wastewater. It employs the potentials of plant roots to absorb nutrients from wastewater. Besides, plant species selected for phytoremediation have the ability to accumulate specific or wide range of pollutants. For this study, an aquatic plant named Limnocharis Flava will be used. Limnocharis Flava is an aquatic flowering which can grow in southern and southeastern Asia. This plant is a non-vegetable in Malaysia. In Malaysia, we call it Keladi Senduk. Limnocharis flower has the ability of absorbing heavy metals as well as pollutant. Planting plants in detention pond in order to sustain the quality of water in it is known as bioremediation of water. Water quality in the surrounding water bodies are improved through nutrient absorption by such type of plants, and this will lessening the transfer of pollutants and chemicals into nearby water bodies. Water that is stored in retention ponds also available for non-potable human users such as irrigation. Placing of plants such as Limnocharis flava may enhance aesthetics. This Limnocharis flava will be used in a detention pond in Politeknik Sultan Idris Shah. So the Politeknik Sultan Idris Shah, PSIS, equipped with a detention pond as a stormwater treatment system. The detention pond also collect water from drain from cafeteria, hostels, and colleges. Since ever water collected, there has become sample for laboratories tests and also students' outdoors activities such as canoeing. Laboratories test normally recorded values of COD at average 100 to 150 milligram per liter and total suspended solid in the range of 0.01 to 0.02 milligram per liter. The detention pond's water is turbid and the water are slightly alkaline. This finding shows that the quality of water needs to be improved because the pond not only acts as a detention pond, but at the same time it becomes a place for outdoor activities and also maintain the landscape of the Polytechnic. Let's proceed to the methodology. Water samples has been collected from the PSIS detention pond and separated into two plastic containers, which is one is for planted with Limnocharis flower and the other container is for the control sample. The total period for this study is 9 days. So the plants were grown over 9 days. Both containers were fixed with a pipe that has a filter and solid may be separated easily through the filter. Water quality were monitored towards remediate water. So there are several parameters that will be measured in the study, which is first is dissolved oxygens, DO, and then the total suspended solid, TSS. We also measure the chemical oxygen demand, COD, turbidity, as well as pH. Let's proceed to the results and discussions. For the first parameter, it is about dissolved oxygen. So based from the figure 1, the graph of dissolved oxygen DO, the amount of dissolved oxygen DO in sample A was 8.17 milligram per liter at day 0, and at day 9, the amount becomes 7.04 milligram per liter. So therefore, 1.13 milligram per liter DO or dissolved oxygen had lost from the sample. In sample B, the amount of DO at day 0 is 7.71 milligram per liter and then becomes 7.19 milligram per liter at day 9. So the difference of DO amount from day 0 to day 9 is about 0.50 milligram per liter. In general, most pond water can hold about 10 to 12 milligram per liter of oxygen. This oxygen is reduced by the biological decay of organic material such as decaying plants and animals or animal and human waste. Dissolved oxygen level below 6 milligram per liter can begin to have detrimental effects on pond life. So based on the analysis data, all samples are above 6 milligram per liter of DO, and this amount is in satisfactory level. Okay, let's proceed to the second parameter, which is total suspended solid, TSS. So based on the figure 2 from the graph of total suspended solid, so the amount of TSS in sample A is 0.111 milligram per liter at day 0, and at day 9 the amount become 0.0035 milligram per liter. So therefore there are 0.1075 milligram per liter of TSS had lost from the sample. In sample B, the amount of TSS at day 0 is 0.0995 milligram per liter and 0.0165 milligram per liter at day 9. Then the difference of TSS amount from day 0 to day 9 for sample B is 0.083 milligram per liter. The high concentrations of suspended solid uh, can cause many problems for stream health and aquatic life. The high TSS in a water body can often mean higher concentrations of bacteria, nutrients, pesticides, and metals in the water. In most situations, a total suspended solid concentration below 20 milligram per liter appears clear, while levels over 40 milligram per liter may begin to appear cloudy. Based on data analysis, all samples had a total suspended solid value that is below 20 milligram per liter. It shows that the pond water had a low concentration of bacteria, nutrients, pesticide, and also metal in the water. All right, let's proceed to the chemical oxygen demand, COD. Based on figure 3, the reading of COD in sample A is 138 milligram per liter at day 0, and at day 9, the amount become 98 milligram per liter. Therefore, 40 milligram per liter of COD had lost from the sample. In sample B, the amount of COD at day 0 is 144 milligram per liter, and the amount become 113 at day 9. So then we can see here the difference of COD amount from day 0 to day 9 is about 31 milligram per liter. According to standard of Central Pollution Control Board, permissible value of COD is 250 milligram per liter. Then based on data analysis, all data are less than permissible value of COD, which is 250 milligram per liter, and it is shows that the pond had a low amount of organic pollution in water. Next is the turbidity. Based on figure 4, it shows that the reading of turbidity in sample A was 166 NTU at day 0, and at day 9, the amount became 19.17 NTU. Therefore, 146 and 83 NTU turbidity had lost from the sample. In sample B, the turbidity amount at 0 day is 238 NTU, and it is reduced to 25.1 NTU at day 9. Total radius of turbidity from day 0 to day 9 for sample B is 212.9 NTU. Turbidity is a measure of how clear the water is. From the data analysis, it obviously shows that there are huge amount of reduction for turbidity in just a period of 9 days. This scenario shows that the use of Limnocharis Flava as bioremediation of water in detention pond can reduce water turbidity. Last parameter that has been measured is pH. Lah. So based on the figure 5, it shows that the pH at day 0 for sample A is 8.17 and reduced to 7.04 at day 9. For sample B, pH at day 0 is 7.71 and reduced to 7.19 at day 9. The pH reading of both samples is in the range of neutral reading. These pH values are suitable for ecology of water. As a conclusion, a Limnocharis flava is a special plant and inhabits wetter tropical environments. It can grow well either in freshwater bodies or slow-moving waterways such as detention pond. Limnocharis flava has the potential of remediating water in terms of dissolved oxygen DO, chemical oxygen demand COD, total suspended solid TSS, turbidity, and pH. The application of Limnocharis flava seems to be less disruptive, economical, and environmentally sound cleanup technology. Therefore, this research has been done successfully because the Limnocharis flava can be used as bioremediation of water that improve the quality of water in detention pond of PCs. That's all from me. Thank you very much.